Today's lesson is on meiosis, and you will learn very important terms that you need to know for your matric exam. Now, before we talk about meiosis, you need to know the introduction and have a little background on meiosis. So we know you learned mitosis, and you learned that mitosis takes place in normal body cells known as somatic cells. Okay? Now, you have to know the definitions of diploid and haploid. Now, diploid is when a cell has two sets of chromosomes, and haploid is when a cell has a single set of chromosomes. The symbol for diploid is 2n and the symbol for haploid is n. A sex cell is known as a gamete, and the female produces the sex cell, the ovum. Please don't use the word egg, so let's scratch that out. It's ovum. And the male produces the sperm. So let's talk about the importance of meiosis. Meiosis is reduction division. Right, so before we talk about that, there's a process called gametogenesis. Gametogenesis is the production of gametes. When it takes place in the ovary, it's called oogenesis, and when it takes place in the testes, it's called spermatogenesis. Oogenesis forms the ovum, spermatogenesis forms the sperm, and it is here that the chromosome number is reduced from 46 to 23, so that the ovum that forms has 23 chromosomes and the sperm that forms has 23 chromosomes. The reason for that is when the ovum and the sperm fuse, it will form a zygote with 46 chromosomes. And that zygote will grow by mitosis into an embryo and then into an organism. So it's very important that you know mitosis and meiosis. Now, meiosis is reduction division. Now, so let's write there. Meiosis is reduction division. What does that mean? A normal cell goes from diploid to haploid. So that's what reduction division is. And the reason for that is when the sperm and the egg fuse, you get a chromosome organism with 46 chromosomes. So it's reduction division to produce haploid gametes. And every time a sperm and an ovum fuse, it forms a zygote with 46 chromosomes. And that is what is our chromosome number of our species. So meiosis also keeps our chromosome number of our species constant. Okay, so that's very, very important. Let's go back to my transparency. So that was the first transparency. Now, these are diagrams for, oh, so sorry. Let me look for my, now, meiosis is made up of two divisions. There's two meiotic divisions. And it's very important that you always, when you're naming the phases, you always put the number 1 and 2. So meiosis takes place in PMET, that's the order, prophase, metaphase, anaphase, telophase. And this that you see as prophase I. And prophase I is extremely, extremely important. Okay, and make sure you know how to spell, and there's a number there, so that's how you would write it. Please don't write just prophase, you don't get marks for it. Now in prophase, homologous chromosomes form. Now homologous chromosomes, let me tell you what is homologous chromosomes. Sorry about the noise, I'm at school. A homologous chromosome, let me draw a homologous chromosome for you. And this is a very important definition, so make sure you know it. So I'm trying my best to draw a homologous chromosome. Sorry about that. but let me just write it P. So a homologous chromosome, if you look at this diagram, this is a homologous chromosome. Now homologous, homo means the same. Now homologous chromosome, can you see I drew them the same sizes, the same shape, the same centromere position. Remember, this is the centromere, and it's very important, it's centromere. And this is a chromatid. Please make sure you know how to spell centromere. So this is the visible reasons why you will say it's a homologous chromosome. Then, there are two other things you have to talk about. Let me just erase this. So same size, same shape, same centromere position, and then same gene. Meaning, on both these chromosomes, we'll have maybe eye color, and then the same gene locus. Locus means the positions are also the same. And can you see one chromosome is paternal, means it comes from the father, and one is maternal. So that is what homologous chromosomes are. So what you see here, in prophase I, homologous chromosomes are forming. Okay? And here, this dotted line here is showing you that that is the nuclear membrane that's disappearing. Disappearing, that's why it's a dotted line. And what you don't have here is the nucleolus is also disappearing. And then you can see there the spindle fibers are forming. Now you can see homologous chromosomes are formed. They're the same size, the same shape, same centromere position, and the gene locus. Then the chromosomes come to lie next to each other. And when the chromosomes come to lie next to each other, they're called a homologous chromosome or a bivalent. That's another name for a homologous chromosome, a bivalent. And then the chromosomes touch. The place where the chromosomes touch is a chiasmata. I'm just going to write there touch. Okay, chiasmata. And then exchanging of genetic material takes place. And this process is called crossing over, and the significance is genetic variation for natural selection. Now, if you look here, you can see this chromosome has some parts green, this has some parts blue, this has some parts red, some parts orange. So it shows you that genetic variation has occurred, and this is crossing over. And this is what allows us to have some of our characteristics of our mothers and our fathers because of crossing over. Okay. So that is now prophase I. This is extremely important. Make sure you know how to draw these diagrams and make sure you know how to do. Okay, so let me just clear. So if you're asked to draw this in class or in your exam, you're only allowed to use your pencil and crossing over only with a pencil. So there, pretend this is a pencil that I'm drawing with, and then you leave the one part clear and the other one you color in with your pencil. That's the only way you're allowed to draw, show crossing over. You're not allowed to use color in your exam. And then if you want to show crossing over, okay, then you will just have this part here cleared. After crossing over, this is what the chromosomes will look like. And then just to show at the bottom, that's how the chromosome exchanged. And one is maternal, one is paternal. So that's actually how you would draw crossing over in your exams. Okay, so that is now the homologous chromosomes, and that is now prophase I, which we're done with. Okay, now the next thing we're going to talk about, so prophase I already has taken place. Very important crossing over takes place. Okay, so sorry about this. Now we're looking at metaphase I. This is one. Okay, it's one. It's one. So 1. Now meta means middle. Now, when you're using the word in meiosis 1, you're always going to use the word homologous chromosomes. So can you see here? The chromosomes have arranged at the equator. Now the middle of the cell is the equator. And we can't call it middle. So that if you look at it, you will see the homologous chromosomes move to the equator and they become attached to their own spindle fibers. So here you always use the word homologous chromosomes. Now, prophase I and metaphase I are the two phases that are responsible for genetic variation. Now let me explain this again to you with my pictures. Now, every time this is taking place in your body, there is no set pattern for the chromosomes at the equator, meaning the chromosomes can be maternal, paternal, maternal. So I'm just going to do this. That's the pattern of the chromosomes. Or, and you get what I mean. The next time this happens in your cell, it could be paternal, paternal, maternal, paternal. It could be like this. That means every time this is taking place at the equator, at metaphase 1 or 2, there is no set arrangement for the chromosomes. And that is what brings about more genetic variation. And that's why I teach you must say random alignment of homologous chromosomes, chromosomes take place at the equator. And then at metaphase 2 also, it'll be random alignment of chromosomes. So it's metaphase 1 and 2 that are responsible for genetic variation and prophase 1. It's very important. And by saying random, it means there's no set pattern every time this happens in your body. Then Anna means apart. Okay. So Anna means apart. So can you see one of the chromosomes of the homologous, one of the pairs, of the homologous chromosomes move to opposite poles. And this is the answer you must memorize. Don't worry about random. They have changed this now in the matric exam. You cannot say anaphase 1 also brings about variation. Okay? So one of the pairs of the whole chromosomes, of the homologous chromosomes, move to the opposite pairs. And that's what happens. And then telophase, the chromosome number is halved. You see, let me just go back to my picture. Prophase. Sorry. You see, we started off with 4 chromosomes. And look at telophase. How many chromosomes do we have? 2. So it's telophase that halves the chromosome number. You started off with 4, and now the chromosome number is halved. That is the purpose of meiosis, to reduce the chromosome number. Now, the next thing we're going to talk about is metaphase 2. I did not draw prophase 2, but I would like to draw it in any case because I want to do a better job and you need a good lesson. Now remember, in prophase 2, it's 2 cells now that are undergoing meiosis 2. Okay? And then again, the dotted line, this is the nuclear membrane. That's the nucleolus. Okay? And then you know your spindle fibers are forming. And then you know we had, and then you had 2 chromosomes, and there's the 2 chromosomes here still in the nucleus. So that's what happens at prophase 2. Now sometimes the cells can completely split in 2 like this, as you will see in your matric exam. Or sometimes, some species, it can carry on splitting and go into meiosis 2. Okay, so that is now prophase 2. Let's move on to metaphase 2. Again, here it's very important. Can you see there's only single chromosomes that move to the equator? And again, the chromosomes move to the equator randomly. So metaphase is also responsible for random arrangement of chromosomes. So single chromosomes move to the equator, and here you can see in this species the cell is still dividing. Anaphase II is very important. Here, the chromosome splits at the centromere, and you have daughter chromosomes that move to opposite poles. Remember, at anaphase I, you had a whole chromosome. Here you have daughter chromosomes. Remember, at metaphase I, you had pairs, homologous chromosomes at the equator. Here you have single chromosomes. And then the cytokinesis will take place, and you end up having 4 cells that form it. And these cells are important. Now, you must be able to label, please. If you asked to label, this will be the nuclear membrane reappearing now. This will be the nucleolus reappearing. This is the cell membrane, and this will be the cytoplasm. Cytoplasm. Oh, it just doesn't give me cytoplasm. Please, can I ask you to know how to draw this stage? It's very important. Most examiners, they ask you to draw this. If you see in some diagrams, the chromosomes are different colors and different shading, please make sure you draw your chromosomes like that. And you can see now, these are the 4 cells that form the gametes. And that is now meiosis II. You started off with 4 chromosomes, and now you ended up with 4 cells with 2 chromosomes each. Okay, and that is now meiosis. Look at the diagrams. Now I want to explain to you how will you identify between a plant and an animal cell. Now, if you look here, you can see that there's these structures called centrioles. And we're only allowed to call them centrioles, no other label. And if they are centrioles, this is an animal cell. And the centrioles form the spindle fibers. So please make sure you remember that if you ever ask to draw an animal cell, you draw the centrioles as well. Okay, so that is now your lesson on meiosis. Now, I still want to talk to you about the significance of meiosis. And the significance of meiosis is, you just learned already, reduction division to produce haploid gametes. You just learned in Prophase I, genetic variation for natural selection. You have that in my junior Taki, so you can look at it. And then prevents doubling effect of fertilization to keep our chromosome number of our species constant. Make sure you know that very, very well. Okay. With mitosis, we also did that the last time you were at the lesson. Produces genetically identical cells, repairs and replaces old and worn out cells, and asexual reproduction in humans. Please make sure you know differences between mitosis and meiosis. And you did do that with me last time. And but meiosis is important because you see what happens in mitosis, in meiosis. Sometimes in a female, when she's old, non-disjunction takes place. And non-disjunction is when a chromosome fails to separate. So instead of this ovum having 23 chromosomes, it has 24 chromosomes and has 2 copies of the 21st chromosome. The sperm will be normal, will have 23 chromosomes, 1 copy of the 21st chromosome. The 2 will fuse and form a zygote with 47 chromosomes, 3 copies of the 21st chromosome, and this is what results in trisomy 21. Okay, and that is now what happens, and this happens at meiosis. Okay, it will be at anaphase I, anaphase II. We're not actually sure. Okay, so let's look at more of our pictures. This is diagrams of mitosis and meiosis, which you can also look at. I want to do this diagram with you. As you can see here, what's happening here? Non-disjunction. This is non-disjunction that's taking place. And it will end up having, so the gametes will have 4 gametes. So one will have, instead of having 1 chromosome each, the 2 will have 2 chromosomes, and these 2 will be empty. We did do this, and in the papers that we did, and this is called aneuploidy. And it's non-disjunction that occurred. Can you see here? Failure of a chromosome to separate at anaphase. So this chromosome is supposed to go this way. And unfortunately, this will be the gamete that will form the ovum, and that's how the woman will actually will have a child with trisomy 21. Okay, I hope that you understand your work. Thank you.